Madam, please proceed, madam.
Okay, sir. Dharani, ma'am.
Please proceed, madam.
Okay, sir. Dharani, ma'am. Good evening, everyone. Welcome to the webinar, detailed seminar on pharmacy project results. Get ready for an enlightening session. Now, I welcome Mrs. Kajilakshmi ma'am, Associate Professor, Vice Principal, Academic, Faculty of Pharmacy, to commence the section with the introduction to the program. Over to you, ma'am.
Thank you, Mrs. Janani, ma'am. Each day comes bearing its gift, untie the ribbon. With this beautiful quote, a warm welcome to respected speaker, principal sir, staff, students, and participants to this national webinar on topic detailed seminar on pharmacy project results. Faculty of Pharmacy, Dr.MGRERI Educational and Research Institute is successfully organizing a series of national and international webinars and FTP with eminent speakers from all over the globe with a vision to intensify the intellectual wealth to the students, faculty, research scholars, industrialists, and pharmacists. In continuation with the events conducted, Faculty of Pharmacy, Dr.MGRERI Educational and Research Institute is organizing a national webinar which is to emphasize the pivotal role and need on discovery of pharmacy project results. The pharmacy project results has exceeded its state global aim of producing 2 compounds at a larger scale and advancing them to preclinical evaluation for infection or central nervous system disease. In total, 5 marine-derived products have been made to animal models for central nervous system disorders, and it is also results in the behavioral model for the Alzheimer's disease, where it results non-toxic and showed drug-like properties. Analogs of these compounds are being synthesized by the US collaborators, and patent protection will be applied by the pharmacy partners and US collaborators. It is also a significant compound to show drug-like properties and non-toxic will be patented by the pharmacy partners involved and advanced to the next stage. In the area of anti-infective, many of the extract containing known antibacterials work will be continued for at least 20 promising compounds in the existing years, as pharmacy also attained its major objectives by reducing the bottlenecks present in the marine biodiscovery pipeline. High-quality taxonomical distinct marine microorganisms are isolated from a range of marine environment, which includes Arctic Sea, Antarctic seas. Improving these techniques for strain isolation, cultivation, elicitization of new compounds has been developed. So it is much interested for all of us to know about the discovery in the pharmacy products. A recorded version of this webinar will also be available in our YouTube channel, named as Principal Pharmacy. You can subscribe this channel for regular updates of the webinars and information. At this moment, I would like to extend my heartfelt gratitude to our management, Founder Chancellor, Dr. A.C. Shanmugam sir, Honorable President, Engineer Thiru A. Ravikumar sir, Honorable Secretary Thiru A. Ravikumar sir, Honorable President Thiru S. Arun Kumar sir. Under those benevolence, all our efforts are taking shape. I take this opportunity to thank our Vice Chancellor Madam, Dr. S. Geeta Lakshmi, for the constant support and guidance. I also welcome and extend my hearty welcome to the speaker of today's webinar, Professor Marcel Jaspars, FRSC, SCD, Marine Biodiscovery Center, Chair in Chemistry, Department of Chemistry, University of Aberdeen, Scotland, UK. Sincere thanks to our beloved Principal Sir for his guidance and encouragement in all our efforts. I welcome all the participants, students, and faculty members present for this program.
Thank you, ma'am. Next, I invite Dr. N. Harikrishnan sir, Professor and Principal of Faculty of Pharmacy, to deliver the welcome address.
Thank you. First of all, my sincere thanks to the Department of Pharmacology, the HOD, Professor Dr. Santosh, and the other department people. So, and the dear management and Dr. Marcel Jaspars, students and faculty members. Good evening and warm welcome to all of you. We are delighted to gather here today for the seminar on detailed seminar on pharmacy, the captivating subject that combines a realm of pharmaceutical and the vast ocean. We extend our heartfelt gratitude to the management for organizing this event, and we are honored to have Dr. Marcel Jaspars with us as our esteemed speaker. Dr. Jaspars' expertise in the field is highly regarded, and we are privileged to have the opportunity to learn from his profound knowledge and experience. Mainly to our students and faculty members, this seminar presents a remarkable occasion, expand our understanding of the pharmaceutical industry and its potential ties to the ocean. It offers unique perspective on the rich resource and untapped potential hidden with our marine ecosystem. We anticipate gaining fresh insights and perspectives that can shape the future of pharmaceutical research and development. We extend our sincere appreciation to all the individuals who have worked tirelessly and behind the scenes of organizing this event, including the organizing committee, volunteers, and our technical team. Your dedication and efforts have made this gathering possible, and we are grateful for your commitment to promoting knowledge and learning. Once again, we extend a warm welcome everyone present here. May this seminar on pharmacy inspire us, ignite our curiosity, and pave the way of new horizons in the pharmaceutical industry. So let us embark on the intellectual journey together and make the most of this enriching experience. Thank you and enjoy the seminar. Thank you.
Thank you, sir. Now I request Dr. V. Santosh Kumar, sir, Professor, Faculty of Pharmacy, to introduce our chief guest for today's session.
A very good evening to all. Good evening to our respected principal, sir, Dr. Harikrishnan, sir, and our today's resource person, Dr. Professor Marcel Jaspars, and all the delegates. Yeah, it's a very interesting profile. So, Professor Marcel Jaspars is currently working as a chair in chemistry, the School of Natural and Computing Sciences, University of Aberdeen, and he holds a degree, a Cambridge degree, university degree, Doctor of Science in the year 2018, and PhD in 1992 from Dublin, and postdoc from University of California, and the external memberships. So Professor Marcel Jaspars with the Scientific Advisory Board, Center for Microbial Secondary Metabolites, Danish Technical University, Denmark, and governing board member of Industrial Biotech Innovation Center, Strathclyde. And some of the previous position is the HOD of Chemistry at the University of Aberdeen and Scientific Project Leader of the European Union FP7 Project Pharmacy, and a visiting professor in University of Tromsø, Norway. And his research involves the discovery of new compounds with potential pharmaceutical use from marine and desert environments, their structure, determination, and biosynthesis. His research specialization is in the marine chemistry, medicine, medicinal chemistry, organic, analytical, applied chemistry. And some of the policy work is a co-author study for the United Nations Convention of Biological Diversity on digital sequence information on genetic resources, and special advisor on marine genetic resources to International Union for the Conservation of Nature at the UN, and co-lead of the Deep Sea Genetic Resources, and member of the Royal Society Group, and chair advisory panel on policy and legal experts in the United Nations, European Union, and NGOs, and UK member of the Biotech. And some of the prizes and awards: Biotech and Biological Sciences Research Council, then American Society of Pharmacognosy, then Fellow of the Royal Society of Chemistry, and Fellow of the Royal Society of Edinburgh. So Professor Marcel Jaspars published around 250 papers. We welcome you, sir. Thank you very much.
Thank you very kindly for your introductions, and also thank you very much for your invitation to speak to you here today. If I could start to share my screen. I don't have the, yeah, there we go, share screen. Screen 1. share.
Yeah, it's coming.
It's coming. That's good.
Yeah, it's good. Looking
Good. Thank you very much. Okay. So today I'm going to give you a description of the background, the origins, and the results of the pharmacy project, which I was in charge of for the science. I wasn't in charge of the administration, just the scientific aspects of the whole project. I'll explain to you the background to where it came from and how we got here. So first of all, often the European Union bases its calls for proposals on reports that have been written. And I was fortunate enough to be part of the European Science Foundation Marine Board paper on marine biotechnology. I headed the health section of the report in 2010. And that was then used as text for a call for proposals. So what happened then is, what you see then is this call here, which in typical European style is a very lengthy title of a description. And it basically gives you a description of what is required. So it gives you the background. It gives you the amount of money they have available. So 24 million is a lot of money, European euros. And they say that more than one consortium will be funded. That means that they will try and fund 2 or 3 typically out of this amount of money. And in this case, the requirement was that only up to 25% of industry was necessary in order for the call to be funded. Now, what happens with these calls is that you have to address every single aspect of the call text. So the call text is about 2 pages. You have to talk about chemicals, pharmaceuticals, biomaterials, cosmetics. You have to talk about processes. You have to talk about marine biotechnology. So it's quite a lengthy application. So it's often several hundred pages by the time you're finished with all the part of the application. So it's quite a lot of effort. Luckily, I was very well supported by my colleagues at the University of Leuven, who were capable of doing all the administrative procedures. So in the end, we were lucky to get funded the next year, 2012. And what you can see then is we decided to call it Increasing the Value and Flow of the Marine Biodiscovery Pipeline. We were funded for 9.5 million euros. We had 24 partners in many different countries, including what is called international partner countries. So in this case, Chile, South Africa, China, New Zealand, and Costa Rica were the countries that joined us on this proposal and were funded through the European Commission. The official description was to improve the quality, volume, and value of active agents discovered in the marine environment and increase the speed at which they can be delivered to the marketplace by addressing bottlenecks and restrictions and adding technical booster pumps. But in essence, we were doing 2 things. We would deliver a product, which was a molecule in an animal trial. And the second thing was to solve some of the problems that were apparent in the biodiscovery pipeline. Officially, we had 4 years to do it. We got a 6-month extension due to issues that arose during the work, which included one company failing during the proposal, having to replace them with another company. We also had issues of illness in one of the partners and a number of other personnel issues throughout. So again, you're managing, as part of this, I was managing the work of about 60 people simultaneously, which is quite challenging given the amount of administration the EU requires. So you can see the partners globally. We had the Institute of Microbiology and the Chinese Academy of Sciences, Wuhan University, University of Waikato, the Institute for Biotechnology in Chile, INBio in Costa Rica, and the University of the Western Cape in South Africa. The other partners are all within Europe. Some of them look quite unusual. There was a company called East Coast, and they were a policy-based company. And the other side, we have the International Union for the Conservation of Nature, which helps a lot with the policy work as well. In terms of companies, we had Deep Tech, we had BioBridge, which is a consultancy, we had Sea Life Pharma, and we had Selecta, another company, again, and Biocom, our communications company. So again, it becomes quite complex when you see it all together. And the point was that we had access to both the North and the South Pole and thermal vents. And the reason for that becomes apparent next. So here's the team that we had. This is the first event that we had. It was in northern Spain. And you can see that already. Our colleagues from Costa Rica were there. Our colleagues from South Africa were there. Whereas South African colleagues who's there somewhere there. Yeah, so it's quite nice to have them all there in the same location talking about the project. And we were very fortunate to have very good scientific advisors, both on the scientific side and the industry side. So we had, for instance, Sheo Singh and Guy Carter, who are industry experts, giving us advice every year at the meeting. So it was really a good support system for us as well. So why pharmacy? Why was it important? The official reason at the beginning was to reverse this decline in approved antibiotics in terms of the timeline. So you can see over the last 40 years or so, we see a decline in the number of antibiotics approved, and we see an increase in the number of multiply resistant Staph aureus infections as an indicator of antimicrobial resistance. So the initial key was to look for new therapeutics for microbial infections and central nervous system diseases, to widen the bottlenecks in the marine biodiscovery pipeline, which I'll enumerate in a minute, to develop mechanisms to transfer marine biotechnology to end users, and to make marine bioproducts more attractive to develop for industry. So what were the legal and technical barriers to success in the marine biodiscovery pipeline? The first one, we were going to be working in extreme environments, the deep oceans and the cold environments in particular. And the issue, first of all, is physical access. How do you get access to deep ocean samples? And we were going to develop a deep sea sampling device that could sample down to 10,000 meters. Unfortunately, this is one of the aspects that was hampered by staff illness. The chief engineer designing this had a very long-term illness and was unable to complete the task. Legal access, we did work on quite a lot, and I'll explain to you at the end of this seminar how we achieved some very important input into the UN process on the ocean treaty. Secondly, in terms of biology, we were looking at different ways to isolate compounds and identify talent in the organisms. Yeah, that was that one there. And then for the chemistry as well, how do we know the molecules are new? What kind of data mining could we carry out? And what kind of tools can we develop to make that happen? In terms of activity, we used a new mechanism of action determinants, so some very interesting tools developed for assessing results, particularly in the CNS activity and toxicity screens. And then at the end of it all, what kind of scale up could we do? What kind of knowledge transfer was essential to make that process attractive to industry? So this was the original discovery funnel. This is what we're going to do. And this was based on experience of the biodiscovery pipeline. So we would try and isolate this many microbial strains. We would have then, each one would be grown in multiple conditions and would be extracted. That would make then about 18,000 different extracts were grown in different conditions. So that's the starting point. Out of this, the success rate would be to profile those using a number of different biological assays, lots of different ones in antibacterial, antifungal, CNS active, and anti-Alzheimer's disease. That would then lead, hopefully, to this many bioactive extracts, which would give rise to 150 molecular families and 2 lead compounds, which would go into animal trials for the next stage of development. So often these processes are very complex. With that many partners, you have to have what's called work packages. These are things that the EU likes to use to make sure that you're doing it right. And we have these pillars, as you can see them down here. So we look at bioresources, we look at generating libraries of extracts and compounds, we look at testing compounds using assays, and then we look at the chemistry isolation, and that would feed back into libraries and then round again through assays to go round and round until we have some pure compounds with activity. There was what's called a demonstration across the way, which meant that you would try and see how you could scale up the formation of bioresources for extraction. You would look at doing, for instance, chemistry at larger scale. We had a policy work package, which I'll explain more about at the end of the presentation. We had a communication package, which was very well handled by a German agency called Biocom, and of course, management goes across all the aspects of the work. And again, management is very highly regarded by the EU. You have to do it a certain way, and you have to do it right. So it takes time to do it, but fortunately, we had a very good administrative assistant called Monica and she helped keep everything straight and make sure that all the information was submitted to the EU on time. So the initial part was to look at strain collections, and in order to get the process started quickly, we realized we needed materials from the beginning. So what we used from the beginning was to have legacy collections from the Arctic, the Antarctic, Republic of Ireland, South Africa, and Argentina. These were samples that were present in the labs taking part in the collections, both from the deep oceans and the cold oceans, and that allowed us to then continue work immediately to do some cultivation under different conditions, extraction, isolation. Sorry, excuse me. The other thing then was to continue with that work to obtain samples from different locations. So the Chinese partners would include mangrove environments, the Arctic from the Norwegian samples, South Africa. I would provide materials from the cold ocean on the southeast coast of South Africa. And we were fortunate enough to have a collection in the South Shetland Trench off the Antarctic. So again, we were able to get samples from those locations and develop new strains. So you can see some of the beautiful strains that were isolated from the Irish collections off the west coast of Ireland at depths up to, I think, 5,000 meters. So here's an example of 3,000 meters. So here's the location, here's Ireland, and we are over here, top right corner here. And what you can see is they collected specimens at 700 up to 2,900 meters deep. They have a remotely operated vehicle. They're able to use live video streaming of the sample when they're collecting. So what they collected was a number of different things. They collected a sponge, 4 different sponges, sorry. You can see their names here at different depths. And they collected sediment as well. And that was used then to use different selective isolation techniques to isolate strains of compounds, strains of organism from there. And you can see again the picture. And what they did was to provide, using 16S analysis of the microorganisms, they were able to determine where they were from. And you can see this wheel here of the compound, the organisms they isolated, and their taxonomic and systematic distribution, which is very important for us to decide where we're going to go and how we're going to carry on with that. Again, through a different scheme called Eurofleets, which uses a redundant time on ships, we'll be able to get access to this thing called the South Shetland Trough. So Eurofleets works by the fact that a supply ship was going from the south tip of South America to the northern tip of the South Pole, Antarctica. And during that period of time, we were able to have the ship for collections. So it went to the South Shetland Trough, which is the world's deepest trench that is also Arctic or Antarctic water. So the temperature is -2 degrees on the sea floor there. So that was very helpful to have those samples for further work and for isolation of strains from that depth. So that's the first work package. The second work package was basically looking at standardized fractionation and generation of extract libraries. And quite often it's quite challenging to see how you would have a consistent process that everybody could follow. Well, luckily, we were joined by a not-for-profit agency called Medina in the south of Spain. And basically they say, well, this is what you do. You take a 10 milliliter fermentation, you shake it, you add some resin, you centrifuge, you discard the supernatant, you extract the resin, and then you transfer it and add water. And then basically there's a whole bunch of steps here, which allows you to get early stage extracts from these very small fermentations and put them into plates, and then also get blanks and against the extract. You can test those very quickly. So as you can see, we generated a bunch of extracts, and you can see that the 2 agencies that were meant to generate the most generated the most. So both the University of Tromsø in Norway and Medina in southern Spain generated a huge number of extracts and fractions, which was very, very helpful for us to work on. So again, that was important. Everybody did their job, and those were all tested against a bunch of different assays. So again, the issue with dealing with this is when you have this many samples, you have that many strains, you have so many extracts, you have each one tested against different particular assays, you end up with a huge amount of data, and it becomes very challenging. So what we had to do is we had to acquire a database. And the only database at the time that could do this is called NAPIS, Natural Product Information System, which allows you to take information from the site, the source, whether it's a sediment or a sponge, to get you the organism, which then is, in our case, a microbe. You generate biomass from that under different culture conditions. You extract the chemistry. If you have a pure compound, it goes into the database, and the activity is associated with that. So it's what's called a relational database. It allows you to connect everything to everything else. So we could, for instance, go activity and find out what kind of activity was shown by compounds from a particular site. A particular organism could be related to the kind of chemistry that you would see. But it's a really critical aspect to any project like this, the data management. And here, in this case, it was hugely important and very well done by Jeanette Anderson in Tromsø. What you can see in the primary screening, you can see the number of extracts we tested, the number of hits we got. I wish I could see the top of the screen. This thing is getting in the way. Okay, well, we can't see what it says at the top there. So yeah, you can see we had some nasty strains, Acinetobacter baumannii. You can see some other E. coli, Pseudomonas aeruginosa, Staph aureus, Aspergillus fumigatus, Mycobacterium bovis as a Mycobacterium tuberculosis, Candida albicans, and then the photomotor response assay for neuroactivity, and then also an anti-inflammatory assay. So you can see lots and lots of assays. So you can see then when we look at the number of extracts screened and the number of actives, you can see that for some things you get a quite high hit rate, like 5 or 6%. Some other ones, the hit rate is very, very low. So particularly for things like Acinetobacter and Pseudomonas, you see the hit rate is about 0.5%. And then for some, far too high. I'll explain how we worked with that later on. But normally an assay positive rate of 2% is sufficient for you to continue with the work onwards. So what you can see here is the idea behind the neuroactivity screening. What was used was an assay called the CNS assay. And here you can see, hopefully, there's no pictures. We have two assays here. The first is what's called a photomotor response assay using zebrafish embryos. You shine a light at them. If they come towards the light, there's no neuroactivity. If you add your compound or your mixture and you flash a light and they don't come towards the light, that's regarded as neuroactive. If the cells die, you know it's toxic. The second assay then is to use this zebrafish assay, the whole zebrafish, and the eyes are tracked using a camera. Okay, so what happens is the zebrafish is made epileptic either through genetics or chemical screen, in this case, using PTZ. What you have then is the fish is tracked, and you can see it from the motion path whether or not at a single concentration it's having an effect on reducing seizures. And then what you do is you do a dose response. So what you get then, here's the assay. You can see the fish moving. Yeah, you can see it's fitting. It has an epileptic fit essentially. Okay, and then what we did then is we started off with 2,000 extracts. We had 300 hits in the first phase of the activity. We then had 97 anticonvulsant hits at single concentration in here. And then the dose response gave us 43 hits out of that lot. Total assay success rate was 2.1%. We checked the chemistry of those 43 extracts. We found about 7 or 8 that were successful, out of which 3 gave us interesting compounds. The other thing we were doing that was really, really important was to look for better chemical informatics to reduce the wasted effort when we find known compounds again. It could pinpoint new compounds, and it could automatically process large volumes of data. The other one was to have an automated structure determination workflow working with advanced chemistry developments in Canada. Again, they were through their subsidiary in Europe, they were working with us on this one. The idea really is to have better drug candidates with new intellectual property. So have a compound that's a new compound that has activity against a particular assay that you can find more quickly. And that's the principle behind it. So we developed, for instance, the Streptomyces database, which was a sub-database of much larger database, but allowed us to search through the data much more quickly and much more effectively. And that was a really good development for us. And that database was helpful for a while. Unfortunately, the software changed with the advanced chemistry development, so that database is no longer current. The other one is a spectroscopic database, which is something we always wanted. You have basically the structure, the mass spectrum, you have the proton spectrum, the UV spectrum. You have a lot of data about the compound. And this was basically compounds that we had in the library or that we bought to put into the system. Again, very useful at the beginning. Unfortunately, this data is now separate from the database because it's no longer working because of the changes in the ACD software. So again, I would recommend you use a different process, which I'll explain with a new project that we're running right now. And again, novelty extraction system. Again, this was working at the end of the project, which was far too late. We wanted it at the beginning of the project. And again, we now have a modern version of this one working in a new project, and I'll explain more about that later. The point is to find novelty in extracts very early on and to couple it with bioactivity. So, I mean, these are the kind of compounds that we found early on. So you can see some of them are family compounds from known families. Some of them are new things. Some of them are completely different from anything that's been found before. So it's quite interesting to see that it's difficult often to find really, really new compounds, although we did find some things that were quite interesting. You can see what we promised in the blue. You can see what we actually found. So again, it's not as high as what we promised, but we nearly hit the target. But what we did have, which was much better than before, was the activity associated with those compounds. We found new activities for known compounds as well, which is essential. Okay, so the issue with all of this is decision making. And because you have a data set containing more than 100,000 data points, you have to be very careful to be able to select your compound. So we had a number of different spreadsheets for this. And we had a prioritization panel that met every month just to discuss what we're doing. And you, for instance, have this, the green means it's done, dereplication is done, scale up, isolation, bioactivity profile is complete. And we would use this in different ways and discussion to try and figure out which compounds to concentrate on for the next stage of scale up, compound isolation, and bioactivity testing. So it was essential to get it there that far. And this is a very human process. You can't replace it at the moment with algorithms, because what it needs is the input of the operators and the users to make sure it all works. So some good results that we had some compounds that were very active against epilepsy. So this was the two compounds that we found. They were from a marine fungus, okay, and they showed a really high activity in zebrafish larvae. We were allowed to progress through to the 6 Hertz mouse model, which is the standard model for epilepsy. And what was useful about this is that you see the model here. You see seizures are almost completely suppressed at a certain concentration of suretin A2, which is the compound at the top. Yeah, and compared to valproate, it's a significant lower dose. So here is 300 milligrams per kilogram. Our compound was working at 10 milligrams per kilogram suretin A2. The nice thing about it is they had excellent ADMET profiles, so they were very useful in terms of possible models for further work. We worked with a group of Jakub Svenda in the Czech Republic to try and synthesize these compounds, and unfortunately, this compound is not active when it's synthetically made. So we're not sure what's going on, but we have a feeling it's to do with the stereochemistry of these two centers here compared to these two centers here. So we're just going to go back and work on that. The second assays were really quite incredible. This was done in Luis Botana's lab in the south of Spain. South of Spain, north of Spain, sorry, south of Spain. And they found two types of activity for this. So first of all, was that this was a very high anti-inflammatory, very strong anti-inflammatory. The second part was that it was strongly it was strongly anti-Alzheimer's as well in animal models. So the issue, the third issue was that it was also immunosuppressive. So the difficulty with a compound that is immunosuppressive and Alzheimer's treating is that it might give a patient, a lowered immunity to disease, which is not desirable when you're talking about treating a patient for a very long period of time. So what happened in in the lab of Dan Romo at Baylor University, what you see is that he managed to make new chromophores out of this material. So what he did, first of all, is basically take the original compounds, simplify them, and then on the one side have activity similar to cyclosporin, but much lower doses. So it's an immunosuppressive. On the other side, he managed to make the anti-Alzheimer's compounds as well. So these pharmacophores were until recently being pursued, but there's some issues at the moment with reproducibility of the biological data. Again, it's quite interesting to see what's going on there. So again, we're working on this still with Dan and with Luis Botana to see what we can do next. So where did we get to? Then I'll talk about the policy side of things. So we promised, a number of different things. 13,000 strains, 15,000 extracts, 90 active compounds, and 5 drug leads. So we promised less than that, and we basically got out 5 drug leads. So we got 2 for Alzheimer's and 3 for epilepsy. There was an additional compound which I did not show for epilepsy, which is actually, again, a very interesting compound. It was looked at by the Danish group at the Danish Technical University. So we feel we've exceeded our original premise of this work, which is incredible. And we got out a lot of good science and continuation projects, as well as incredible policy influence. So basically we have 130,000 screening events and lots of activity, which we can continue with on subsequent projects. Dissemination was very positive. We had a huge media coverage, especially since the BBC linked us directly to the antimicrobial crisis the day after a major speech by the chief scientist for the government, Dame Sally Davies. So again, we got 24 million hits to our website the next day, which was incredible. And we did have a lot of influence. So we had CNN documentary made, we had an Al Jazeera short clip made for our work, and a whole bunch of social media. And we won several awards for communication, as well as inclusiveness of staff around the world. So I'll tell you more about the projects that followed on, because they're quite incredibly, basically once you have one of these projects, you often have a good working core staff that you can work with. So first of all, we had this project called a RISE, which is a student and staff mobility, which included international cooperation partner countries, allows you to move staff around the world, essentially to have experiences and students to go and have secondments abroad. The second one was an innovative training network called MARPIPE, which were using the samples that came out of the pharmacy project. So that's basically the same idea, but this time looking at the next generation of marine biodiscovery scientists. And again, we had partners in South Africa in this one. The next one is the Marbles project, which is led by the University of Leiden in Holland. And it's looking at the same kind of thing, but much more focused on aquaculture and crop diseases. More recently funded, we have the doctoral training network HotBio, which again is looking at marine biodiscoveries holistically. And we just have our student started, who's from India, Ria Desai. She started with us only a week ago, I think. What is it? Yeah, a week ago. And we have a project called Blue Remediomics, which is 9 million euros, looking very intensely at the marine microbiome and using a whole pipeline of biodiscovery all the way from genetics onto expression and production. So a little bit more detail. So the Marbles project has, again, work packages. What you can see now is biodiversity mining, disease suppressive microbial consortia, and omics-guided awakening of cryptic biosynthetic gene clusters. Is basically the microbial side of things. And on the chemistry side of things, we have small molecules, generation of chemical diversity, and screening and validation. And then on the other side, we have societal aspects, dissemination, and project management. And you can see here, marine biodiversity goes to active agents, goes to production, and goes to application testing. And we're at the stage of having some very unusual strains of bacteria where we are finding activity. Early in the project, we managed to have one of the partners develop a new toy, a new tool, essentially. And what you can see here is this is called Pharma. You can find it if you just type in Pharma and bioarchive. It'll give you the publication as well as a link to the software itself. Now, it's pretty incredible because this does all of the things we wanted to do during the pharmacy project. It allows you to compare directly different extracts from organisms and compare the biological activity against the bioassay data. And it allows you to stream the information from a very large data set to a very small data set very quickly. I think it's an incredible achievement. That's Micha Zjuk in Wageningen in Holland, working with Justin van der Hoofd and Marnix Medema. And they have developed this tool. It's available online. We type in Pharma, and I said bioarchive, you'll find the paper, and you'll also find the link to the software itself. And you can try it out if you want. So I think it's a pretty incredible achievement because what it does, it allows you to select things for novelty, how complex these mixtures are and the bioactivity, and again, how much you think is in there. This is the Blue Remediomics project, which is the other project that we're part of. This is much more about databases and cultivating, looking at databases, looking at strain collections, looking at how to analyze those data and then culturing things at different scales, and then working on getting new enzymes and new chemistry out of it. So our work on this one will be against on antimicrobial peptides. And again, it's a very good team. We have some very top scientists involved in the aspect of biodiversity, genetics management, so gene databases, GenBank, etc. Again, the project that you can see, again, is composed in the work packages, slightly different: bioinformatics, culturomics, processes and products, aquaculture, and environmental services. And then the nice thing about this work package 4 is that what it does, it also includes something called an ocean microbiome health index, which is something desperately needed to try and figure out the health of the ocean microbiome. And then we have a policy work package, communication work package, and a management work package as before. The HopBio project has a less difficult structure. It is basically work package 1, networking and training, coordination and management, and then we have microbiology, chemistry, bioactivity, synthetic chemistry, ADMET, and then training courses from lab to society. So again, it's you can see a pattern emerging that these European things want to see some production, some knowledge generated, knowledge applied to society, and some kind of policy aspects to it to make it successful. So let's talk a little bit about the policy aspects then. I'll just have to go back to some explanations of what we do on this stage and why it's important. So basically the ocean at the moment has a number of zones. And what you can see is you have the territorial sea, then you have this exclusive economic zone, and beyond which you have the high seas and you have the seafloor. And unfortunately, the lawyers decided that the high seas are subject to one legal regime and the seafloor is subject to another legal regime. So what they call the high seas is subject to freedom of the high seas, meaning that if I catch a fish there, it's mine and I don't have to share it with anyone. If I collect something from the area, the seafloor, it's subject to common heritage of mankind, meaning I have to share it with everyone. So if I find something here, I have to share it. At the moment, legal regime is that below 200 nautical miles, we have the Convention on Biological Diversity and the Nagoya Protocol, and India is one of the most advanced nations in terms of having a system in place to deal with this. Beyond this, the United Nations Convention on the Law of the Sea applies, and marine biodiversity is currently not protected, and that's about to change. I'll tell you what's going on there. So sorry. Within the EEZ, a country's jurisdiction, it belongs to you. It's a sovereign property of that country. And then what we deal with is the Nagoya Protocol, and any collections must be with prior informed consent and the mutually agreed terms and with equitable sharing of benefits. Not all countries have ratified this or have mechanisms in place to deal with it. So what you see then is the rest of the bits that are covered in brown are the exclusive economic zones. What remains is about 60% of the ocean and 40% of the planet's surface is not governed by any legal regime that protects biodiversity, and that needed to change. So what we have is, in the areas beyond national jurisdiction, the UNCLOS applies, and that's for the peaceful uses of seas and oceans, it says. It's really important to understand that that's, there is that statement that it is for protection of the planet, but there's no mention of what biologists, what the lawyers call marine genetic resources. They only refer to resources which are defined as non-living mineral resources, which you can see, the two conflicting elements that I mentioned, the freedom of the high seas, has the freedom to conduct scientific research, and then on the other side, that requires sharing of results. And the other side is a common heritage of mankind. No state shall claim sovereignty over any part of the area or its resources, meaning you couldn't officially patent a discovery made from the seafloor beyond national jurisdiction. Okay, so what happened? In 1982, the UNCLOS excluded marine genetic resources, but allowed freedom of scientific research. In 1996, there was an article called The Deepest of Ironies by La Gloca that raised the issue. And that was basically saying that deep sea biodiversity had been excluded from the treaty, and therefore there was no explicit protection for them. In 2000s, early 2000s, they started a very long titled working group. It's called the Ad Hoc Open-Ended Working Group to study issues relating to the conservation and sustainable use of marine biological diversity of areas beyond national jurisdictions. They ran for more than 10 years. And in 2015, when we were part of the Pharmacy project, they just made a decision to develop an international legally binding instrument under UNCLOS. And we were lucky and to be just in the right place at the right time to provide scientific advice to this. So in 2016, they started the preparatory committee, which is open to all UN member states. But what they do is they develop what goes into the treaty. What are the elements of the text? Not the text itself. It just says, these are the kind of things we will discuss next time. In 2018, they started an intergovernmental conference to consider recommendations of the preparatory committee. That was decided. It only started in 2019. And then we'll describe now what happened next. So it was called the package deal. And what that meant in particular, that these things are treated together and as a whole. And this is the bit I'm interested in: marine genetic resources, including questions on the sharing of benefits, with area-based management tools, environmental impact assessment, and capacity building and transfer of marine technology were the other aspects of the treaty. The issue with this one here was the one that was most likely to disrupt the proceedings, because it was the one that was the most scientifically complex and had the possibility of making challenge or trouble for the rest of the treaty if it wasn't completed. So the issue then is that Nagoya Protocol is bilateral. Again, it's just between two countries. The BBNJ treaty, as it's called, is multilateral. So every country has access to the materials in the ocean, and somehow there has to be a process by which that area can be governed effectively using a multilateral instrument, as well as having the potential for sharing any discoveries that are found on the seafloor, and if they raise any monetary value as well. So we created a science policy interface. We brought together scientific practitioners from research and industry. We brought together legal experts and policymakers from different panels, from the EU, from the United Nations, and other secretariats. We provided policy workshops. We provided awareness raising by writing articles for the scientific community. And we shared best practice through a toolkit that we generated for people to access and look at in order to know what they should be doing. This ran for 4 times over 4 years and was incredibly successful in putting people together to explore ideas openly and freely. We also ran workshops at the United Nations. This is my first time. You can see I'm not looking particularly excited about being there, but it gets more exciting as we go along. This was our host. It was the International Union for the Conservation of Nature. Really, we had a very good guidance from Thomas Greiber, and we had good partners as well from different agencies. So the way that you work into the process is you either write scientific briefs, you can have a text provided ahead of the treaty. We often do this online now, so I've taken part in 4 of these now. And you can make a statement on the fly in the room. So again, I've been fortunate enough to speak at least once in the chamber to discuss potential statements supporting the scientific aspects of this. So, I mean, you can see the kind of events we got involved in, and I think up to date now I have more than 50 of these events where I've taken part, provided advice as a scientist, and helped develop policy options. So the kind of things we provide information on is kind of diversity of marine genetic resources, good practice in marine bioprospecting, examples of successful exploitation of marine genetic resources, the kind of benefit scenarios, what's possible in terms of traceability, I'll just say more twice, capacity building ideas, and scientific advances that might affect implementing the agreement. A main paper that came out of the discussions with the working group, the advisory panel of policy and legal experts, was this paper here, which was led by Arianna Broggiato. And we all got involved as scientists and policymakers and everything else. And we talked about balancing governance of marine genetic resources in international waters. And the key output of this was the fact that the current draft of the treaty, which is the one that will hopefully be adopted in 2 weeks' time, contains core ideas that we generated during our work. So I'm very pleased about that. And the other part of this is that Arianna is now the chief negotiator for marine genetic resources for the EU, which is very helpful to have her in that position. So one of the ideas that we had that's still in the treaty is this idea of the obligatory prior electronic notification, that before you go on a research cruise, you let other people know through a centralized system that you want to be going out, that you're going to go out on a research vessel to collect specimens. If anybody would like to join or would like some of your collections, they can let you know at that point. When you get back from your research cruise, you update your electronic notification and you indicate what you have collected. So you collected 50 sediment samples from 5,000 meters deep. You collected 6 sponges and 2 sea cucumbers. You might want to inform people about that. And that allows people then to say, well, actually, I would like a little bit of sediment. And please can I have access to some of the raw data you generators again. At this point, this is something that's not included. There's an exclusivity period. For instance, if you did some work and you wanted to tell people about it, you could publish it, or you could patent it. If you patented it, you might have to pay a small fee to the system in order to enable you to continue with an embargo. And then this is the more contentious one. If the implementing agreement requires monetary benefit sharing, royalties are to be paid on commercialization at a fixed % by sector. This is still being, there is something like that in the treaty right now, and that's being discussed right now. Well, not right now, after the 20th of June, that'll be started to be discussed formally. So what are the benefits? Again, in order to sell this kind of treaty to scientists, there has to be a reason for that. So again, this kind of open mechanism, which is not, which is basically an electronic notification, means there's open access to raw data and samples. It standardizes data management, taxonomy, and species identification, and enhances international research coordination and cooperation. Again, these are the positive things for scientists for a little bit of extra admin stuff. So again, we provided a toolkit, as I mentioned before. This is very helpful. This has now been taken over by the Flemish Institute for the Sea. So what we had so far. So, I mean, the kind of advice that we provided, wish I could see what it said. Oh. Anyway, we provided definitions. We highlighted and agreed on elements of good practice. Basically, the questions we asked at the beginning were reporting on notification procedures. How will it be monitored, policed, and by whom? Yeah. Okay, so who will be in charge of traceability? And again, that's a different system now. How can we manage expectations for financial returns? Can we make sure an implementing agreement is flexible enough to cope with scientific progress? These are issues that are still up on the table, essentially, because a lot of this will be dealt with not through the treaty itself, but through the conference of the parties and the scientific and technical body and the benefit sharing committee. So this light touch regulation is something that seems to have come about now. So what happened in the last couple of years is, of course, the pandemic. That was supposed to happen in March 2020, happened in March 2022, but was closed to civil society. That means people like you and me, scientists, were not allowed in. Then they had another meeting in August 2023 when we were all allowed into the room. There was an attempt to finalize the treaty text on the Friday night. It did not succeed. And the The main issue was, as I predicted, the marine genetic resources. They did not have definitions. They had this new issue called digital sequence information, which came from the Convention on Biological Diversity, which is essentially DNA sequences. How were they going to be included? We had monetary versus non-monetary benefits. What to use the monetary benefits for, fish to be included or excluded. How do you use the notification system, and how do you have identifiers? Traceabilities of materials and data, financial mechanism, and other issues were mainly legal in nature, dispute resolution, decision-making, procedure for adoption. But this is still challenging. So they had another meeting in February 2024, and I was fortunate enough to go to the second week of that. So on the end of the Friday, it looked very bad. They were stuck on this issue of fish. They had no text agreed. On Monday, there was little progress. On Wednesday, the 1st of March, the president of the process developed a text based on all proposals, a compromise text. It was rejected by the G77, but there was internal dissent. What I hear from my colleagues is that not all of them agreed to rejection. EU then developed a text the next day, and now it starts to get difficult because this is the last day of the treaty, Friday the 3rd of March. Right? They started at 9 a.m. Many delegates have only been sleeping for 4 hours. As a scientist, I was in the room until about 6 o'clock, and the last questions from the scientific nature got asked at about 5 o'clock. At that point, the remainder was political negotiation. They carried on throughout the night. They carried on all the next morning. They were kicked out of the room. They had to go to a different venue. And I had to leave at 3 o'clock in the afternoon because my plane left at 7. So I left the United Nations at 3 o'clock. And they continued. And I woke up in London to find out that the treaty had been agreed in principle after 38 hours of negotiation without sleep. Quite incredible. So what does it mean? Here it is, the text that was agreed on the 4th of March. It says, what's happening now is they're doing legal scrubbing. That means they're checking consistency internally and externally. And hopefully on the 20th of June, they will adopt the text, which means it can move towards ratification, which is the next stage in the process. So hopefully there's a coalition of a high ambition coalition that wants to get this adopted within 2 years. And again, that would be very good for the oceans to have a system in place to conserve it and sustainably use it. Yep, it'll take a little time to figure out what's going on. I'm working with a group of global researchers to try and write a brief text on what this means for scientists, essentially. So we should have that out early next year. So what is the good thing about it? Well, a lot of it's consistent with the Nagoya Protocol. The positive side is that the benefits, if any monetary benefits arise, they'll be used for conservation and sustainable use. Fishing is excluded, except where you use it for marine genetic resources. There will be notification. You're given a standardized batch identifier to do with the cruise that you can relate to your samples. You should deposit your samples and data. There will be benefit payments into a fund based initially on assessed contributions per country and other payments later. And then there's the reporting of use of samples and data later on, and inclusion of the DNA sequence information to work together with other treaties. And there will be a benefit sharing committee. So again, it's a very positive thing for science that this has happened. It's very positive for the ocean to be able to conserve it over the next generations of humanity. So I'd like to thank all the partners that were involved in the pharmacy project and the subsequent projects as well. And again, we acknowledged massively the funding that the EU gave us for this work. To get such a highly functioning consortium that had not only effects on scientific discovery, but also global effects on policy development to preserve and conserve the oceans. Thank you very much for your attention, and I look forward to your questions.
Thank you, sir. Thank you for your wonderful talk.
Thank you.
Now I kindly request to accept the e-certificate on behalf of Faculty of Pharmacy.
Thank you. Thank you very much for that.
Thank you, sir.
Wow. Appreciate that.
Now I welcome Mrs. M. Devi ma'am, Assistant Professor, to take over with a Q&A session followed by vote of thanks. Over to you, ma'am.
Good evening, sir. Thank you for your informative and valuable talk, sir. We have received some queries from the participants, sir. In that, the first question from our principal, sir, whether biological active compounds isolated from marine sources having diverse biological activities like acting against cancer, infectious disease, and neurological disorders.
So what was the I missed the question. What was the question?
Our principal, sir.
Oh.
This is the question from our respected principal, sir.
Madam, please repeat that question or
sir, this is
type
the
it in the
question
chat
from,
box.
Yes, sir. So this is the question from our principal, sir. Whether biological active compounds isolated from marine sources having diverse pharmacological activities.
I mean,
like anti-cancer activity.
Yes. So normally, I mean, the ones that we know very well are particularly for cancer because the field was at the beginning in the United States, was funded heavily by the National Cancer Institute there. So most of the drugs that are now in the clinic are for cancer treatment. So there's about 17 or 18 drugs in the clinic now from the marine environment. And I said most of those are cancer, some pain for pain from the cone snails, some antivirals, and now also for heart disease. So again, it's a few drugs in the clinic now from that. Again, diverse activities, because we're testing now for many more different things. So we often find the same compounds that were inactive for cancer are active for, in this case, Alzheimer's, or maybe they're active, first of all, they weren't active for epilepsy, but they're active for antibacterial agents. So the difficulty is there's no way to predict what the activity will be at the beginning. So you often have to just do a screen, and then you filter down to the right level.
Thank you, sir. This is the query from Dr. S. Nandakumar, sir, Vice Principal of Faculty of Pharmacy. How significant was the results of anti-seizure activity on zebrafish model when translated to mammalian models, especially with azapiroviral?
So there were two things there. One of them is that the zebrafish model gives you additional information on the activities. So first of all, it gives you whether there is liver toxicity or whether there is whether it goes to the brain. So it tells you a lot about the administration. So at the beginning, we did cardiotoxicity and neurotoxicity, cardiotoxicity and liver toxicity in the strain, in the zebrafish. The second one was that we were able to determine whether or not it would go to the brain because you have good admin properties. So it's a very good model for translating to animal models. So out of the ones that we took from the zebrafish to the mouse model, I think most of them were successful. We only had like five choices, five compounds that we tried, and I think 3 of the 5 were capable of going straight into the animal model and have an effect. So they not only could they be administered, they could go through the blood-brain barrier to the brain and have the anti-seizure effect. So it's quite a good model. Again, both for counter screening to make sure that they're not toxic, and the other side for giving you information on whether or not it goes into the system and whether it's excreted very quickly. So ADMET is almost guaranteed with this.
Thank you, sir. This is the question from Mrs. S. Gajalakshmi ma'am, Vice Principal of Faculty of Pharmacy. Time to time, marine species varies. How we will carry research work on marine product if we need an excess quantity? How we can get the same species, sir?
Okay, so quite often what we see with these things is that the initial isolation is only a few milligrams, and the yields are quite low as well. So if you use a macroorganism, It's definitely difficult to get enough material, and typically you would try to find alternative mechanisms for production, and chemical synthesis is one of those. The famous examples where people have actually made drugs that are in the clinic right now, it's either synthesis, like a compound called haloven, or it is semi-synthesis, which is something similar to a compound called yondelis, where they take a fermentation product, pure compound that's isolated, and they make chemical synthetic steps on that to get the final product. If you have the microorganism available and it can produce at high levels, then you could do fermentation optimization. And that was the case for salinosporamide, which is in clinical trials still. And they were able to produce a kilogram of pure compound using fermentation. But in other cases, when the yield from the fermentation is too low, you have to think of using recombinant DNA methods to try and express it in another host. And that's not really been done except for non-marine compounds. So a compound called epothalone is a derivative of that that's made using a recombinant method by a company in California, I think. So again, there's different routes to get enough material, but it's not common to go back to the sea to collect materials.
Thank you, sir. This is the queries from Dr. S. Siddharth, sir, Associate Professor. Apart from bacteria and fungus, any actinomycetes have been found during the isolation of marine extracts during your study?
Yeah, I mean, we tend to focus on actinobacteria because they are successful. We tend to try and focus on the rare ones as well. So, you know, if we try not to just focus on Streptomyces, which is really the one where most of our antibiotics and many of our cancer agents come from, we tend to try and go for more remote families of organisms in the actinobacteria. On the other side, people have started looking at unique niches. So there is a group of bacteria from sponges right now called the Tectomicrobia, which were discovered by Jörn Peel in a sponge, a deep sea sponge in, oh God, goodness knows where it comes from. I think it comes from Japan. And that is incredible because that Tectomicrobia is a whole new phylum of bacteria, and they have a huge number of biosynthetic pathways. So again, it's not culturable yet. So what they've done is it's all been recombinant expression of the pathways in a different host.
Yes, sir. Thank you, sir. This is the question from Dr. Evelyn Sharon Mum. Is it possible to mix plant extract with marine extract to evaluate combined activity?
So, I mean, this is the idea of synergy, right? So, I mean,
Yes, sir.
my feeling is that it's difficult to do adequately, but on the positive side, a lot of the different regulatory agencies now allow extracts to be approved for treatment. And there's a famous example of a green tea extract for benign tumors in the United States. And the key with that is if you want to mix extracts for combined activity, you would need to be able to prove that they are the same mixture each time. You don't want to define all the components. That makes it challenging for regulation. But if you can say, without a doubt, that using metabolomics, you can prove that the mixture is the same composition each time, then you could go for approval. It is very difficult to do. I don't fully understand that, but there are groups that specialize in this kind of work.
Sir. Thank you, sir. This is a question from Dr. Jivian Kiranmayi Ma'am, Associate Professor. What are the possible side effects that may arise if the marine source-based drugs used for long term?
So what's the possible side effects of what?
What are the possible side effects that may arise from the marine source if you are using for long term? Marine-based drugs, if you are using for long term, what may be the side effects may get arrived?
I'm not sure I understand the question.
Okay.
Question. Yeah, I'm not following. What are the side effects of what exactly?
If you are using marine-based drugs, if you are using in long-term means, what are the possible side effects?
I got it. Sorry, I got the question. That's hard to say. I know that for a lot of cancer drugs, the side effects are usually tolerated because they are so important. So, for instance, for Yondelis, there are some kind of side effects, which I know exist. But given that it's treating a type of cancer for which there's no other treatment, often patients are willing to put up with the side effects. Yeah, it's hard to know what other side effects you might have. So it's most of these drugs at the moment are cancer, so they're short-term indications. They might be like for a few months maximum. So it's challenging to get something. There's no drugs from the marine environment that are for long-term treatment yet. So we might be seeing that in the future, but the same with any other side effects for long-term uses of a drug. You have to be careful that you report the information through whatever system you have nationally.
Thank you, sir. This is the question from Dr. M. Sudhamam, Associate Professor. What are all the basic criteria for the selection of marine strains that collected under the sea?
Wow, okay, that's a really good question because that's what we're working on right now. So we tend to use 3 filters, as we call them. So the first one is taxonomic diversity. So we're looking at genetics to start with. And initially we started with something like 16S, which allows us to give some diversity analysis of the things that we have. So we might then secondly look at the chemical diversity. We use metabolomics for that. So we take an extract, we put it through the liquid chromatograph mass spectrometer, and we do some networking analysis. And the tool that we've just developed, Pharma, helps us prioritize that. In addition to which we will need the bioactivity data across a number of different screens. So you can find the 3 types of data can be combined to select the best strain, right? So you can basically do a crude extract, maybe some fractionation, you test the activity, you look at the chemistry, and you use those criteria to filter down to the ones you want to work on. What we do at that point quite often is to do cultivation under different conditions. So we have what's called the one strain, many compounds approach, where you cultivate under many conditions and you find a lot of compounds being produced. And what we would do then is, if it looks good, we would do the whole genome of the organism. And at the same time, then we can look at whether or not what we expect the organism to produce is actually what is being produced under those different culture conditions. So it is quite an iterative process. We're getting better at it all the time. We're getting better at refining and finding things more quickly. That's the most important thing, that we get a better success rate. We might not always be perfect, but if we can go from 2 to 3% success, it's already a major success for a project. So that's what we're aiming for in the Marbles project.
Thank you, sir. This is the last question from Dr. S. Anusha Ma'am, Assistant Professor. What are the challenges have been faced by the researchers while doing research on marine sources?
All kinds of things. That's a big question. Quite often, the challenge that we face is the beginning is getting access to collections. So it's knowing where they are, who's got them, who's going out. So it's a physical access. Legal access we're working on. And the most important thing I would say at the moment that we have challenges with is the differences that marine species have with the terrestrial species. So we often find it difficult if we have a macroorganism like a sea sponge or something. There's very few people capable of identifying them adequately, and those people that do exist are overworked. They have too many sponges to identify. So that's a major challenge. On the microbial side of things, we find some very rare and unique bacteria that are very difficult to grow, maybe, but also difficult to once you find out how to grow them or how to sequence them, it's difficult then to get them to produce that chemistry. So again, it's this issue of knowing how to grow certain bacteria. And then the next part is genetics. You may find that an organism has unique genetics, and it's very difficult to figure out what the chemistry is that it produces. So a lot of novelty means a lot of difficulty often. So those are the major challenges I would point out. So both on the one side for access, then for sort of spotting chemical talent and working on from there.
Thank you, sir. Thank you, sir, for giving your elaborate explanation to clarify the queries of the participants. Thank you, sir.
Thank you. Thank you for your attention.
Thank you, sir. Now let's move on. Vote of thanks. On behalf of Faculty of Pharmacy, it's my privilege to deliver the vote of thanks. First and foremost, we extend our heartfelt gratitude to our management, founder, chancellor, Dr. A.C. Shanmugam sir, Honorable President, Dr. S. Arunkumar sir, Honorable Secretary, Thiru A. Ravikumar sir, under whose benevolence all our efforts are taking shape. Extend thanks to our Vice Chancellor, Ma'am, Dr. S. Geeta Lakshmi, for the constant support and guidance. I'm extremely thankful to the speaker, Professor Marcel Jaspars, sir, to grace this occasion with this valuable and information talk on pharmacy project results.